And as I've already mentioned, some individuals should not be exposed to fluoride. People with poor kidney function, people with low iodine intake. Why aren't, and people that work in mines in Western Australia who drink 10, 11, 12 litres of water a day, these people shouldn't have fluoridated water? These people are not being warned. No steps are being taken. How ethical is it to say we want to help some individuals in society, particularly children from low-income families who have higher rates of tooth decay than children from high-income families? We want to help those children. I agree. We need to help those children. This isn't the way to do it, but we need to do that. But why? How can you possibly, from an ethical point of view, say we're going to help these children at the same time we're going to make life worse for people with poor thyroid function, people with low iodine, people with poor kidney function? That's not ethical. It's not ethical at all. No agency accepts responsibility for harm if and when it is demonstrated. Who has the liability for this? We will only find out when the lawsuits begin to happen. The health agencies in the fluoridated countries are doing a lousy job of doing even the most basic studies, most basic health studies. And what is happening now, most scientists in the world are having to rely on studies done in countries like India and China, Iran, Mexico, where they have high natural levels of fluoride, where they are trying to get the fluoride out of the water, and they are genuinely interested in doing some science to find what levels actually cause harm. That's where the science is being done. It's not being done in the fluoridated countries. And so the conclusion that you come to is very, very disturbing. It does not make me happy to say this. The evidence is that in Australia, as it is in New Zealand, as it is in England, as it is in Ireland, as it is in Canada, as it is in the United States, it's more important for our health agencies to protect this policy of fluoridation, for whatever reason, than to protect the health of their communities. Okay. By the way, when we talk about the fact that some people are sensitive to the toxicity of fluoride, do you know something? That's exactly what you expect. Any toxicologist will tell you. The response to any toxic chemical in the human population goes through a bell-shaped curve. What that means is a bulge of the bell. Most people have an average response to that toxic substance. But down at this end, there will be some people who are very resistant and can be exposed to maybe 10 times more of that toxic substance without showing any effects. That's one end of the bell-shaped curve. At the other end of the bell-shaped curve, there will be some individuals, a small percentage, 1 or 2%, who are particularly very sensitive to this substance, and will experience the same symptoms that we see with people that are exposed at high levels in industry or in these naturally occurring areas in India and China. You'll see it in these people. So it's exactly what you would expect. A small percentage being very, very sensitive to fluoride. But that's denied by every fluoridating country. And there's another shocking bit of irresponsibility here. And that is, we've known since the very beginning, the severity of dental fluorosis, which is a mottling and discoloration of the teeth, which is only caused by fluoride. That's why it's called dental fluorosis. It means poisoning by fluoride of the teeth, dental fluorosis. The severity of that condition reflects how much fluoride the kid was exposed to before their permanent teeth came out. And if you're an epidemiologist, it's a wonderful tool in epidemiology to have a biomarker of fluoride exposure, which is non-invasive. You can just look in the kid's mouth, see the severity of dental fluorosis, see how various childhood complaints, etc., early onset of puberty, lowering of IQ, bone fractures in children, etc., see how they relate in your population to the severity of dental fluorosis. Never done it. Never been done in Australia. Never used the severity of dental fluorosis to indicate if there's any relationship to any childhood complaint. So, there is, again, going back to this, all of this, despite the fact that there's no scientific reason to swallow fluoride. No one has ever said to you or me, here's why we need to swallow fluoride, because it goes to this organ and helps to do this X, Y, and Z. And you're probably now wondering, well, what about the teeth? Well, what they've admitted, and I'll come to this in a moment, what they've admitted to is that the major benefit of fluoride is topical. It works on the outside of the tooth surface, not from inside the body. So that's why you can have something that may have an effect on the teeth, but it's not a nutrient. It's not an essential nutrient. Okay, so I'm actually coming to that now. Fluoridation is unnecessary. In 1999, the Center of Disease Control, which is the #1 promoter of fluoridation in the United States, let me qualify that, it's the Oral Health Division, that's 30 dentists, essentially. 30 dentists out of 20,000 employees at the CDC promote fluoridation. And in 1999, they produced this self-serving document which has been used countless times. This is the document which underpins the claim that fluoridation is one of the top public health achievements of the 20th century. If I had $1 for every time a journalist, an editor, or a politician quoted that, I'd be very, very wealthy indeed. But in this paper, they did at least admit one thing. They said, we used to think that caries preventive properties were due to the accumulation of fluoride in the enamel of the teeth during development. In other words, under the gums. Under the gums, the fluoride was accumulating in the enamel so that when the enamel emerged into the teeth, they would be more resistant to acid attack, which is the first step in tooth decay. That was the theory. But this is what they said. However, laboratory and epidemiologic research suggests that fluoride prevents dental caries predominantly after eruption of the tooth into the mouth, and its actions primarily are topical. Again, when they admitted that, that should have been the end of fluoridation right there. Because if you don't need to swallow it, there's no need to put it in the water. And when you've got fluoridated toothpaste, universally available, designed to brush directly onto the surface of the teeth at a much higher concentration that you're ever going to get from water, then this is a sheer nonsense. But again, it underlines the fact that they're protecting this policy and not us. I've said that. And I've said this. Why swallow fluoride and expose every tissue of the body, including the baby's brain, to a toxic substance when you can brush it on your teeth and spit it out? And why put it in the drinking water and force it on people that don't want it? In other words, in 1999, they had the solution to the medical problems caused by fluoride and the ethical problems of fluoridation. Right there, they could have just switched to topical treatment as being the #1 thing that they could promote. And we'll see later, this is exactly what Scotland has done. Fluoridation is unsafe. There are many biological processes that are harmed by fluoride. I would go as far as to say that biology is incompatible with fluoride. It's very difficult for very delicate biological mechanisms to exist side by side with the fluoride ion, and I suspect that's why it's not in mother's milk. Fluoride inhibits enzymes, it switches on G proteins, and does many, many other things besides. Now, bottle-fed babies in fluoridated communities are exceeding the EPA's safe reference dose. So that's what the EPA determined. The safe reference dose is 0.06 milligrams per kilogram body weight per day. When you're getting down to a dose which you say is safe, and you want to apply that to the whole population, you've got to take body weight into account, because it's the same dose. It's going to be far more harmful for a baby or a young person than an old person. So we normalize it by dividing by the adult's body weight. So you find the safe dose for an adult, then you divide by the weight of the adult to get the dosage that you can apply to the whole population. So that's the safe dose. Now, in 2006, we had this very important report from the National Research Council, 2006. This is the council that I testified before in 2003. It was a remarkable event, this National Research Council, because this was the first time in history that a truly balanced panel looked at this issue of fluoride's toxicity. They had pro-fluoridation people, 3, they had anti-fluoridation, 3, and they had 6 who hadn't declared their position. So balanced panel. They expected to take 1 year to review the literature on fluoride's toxicology. It took them 3.5 years, and eventually this came out. It came out 1 year before the National Health and Medical Research Council produced its last review of fluoridation. Remember I was talking about rubber stamps? In the NHMRC report, they dismissed this NRC report, which was incredibly thorough, and made their review look pathetic. Most of it was cribbed from the York Review anyway. But they dismissed it in one sentence and said, readers should be aware that the levels that they looked at in America in this report were much higher than the levels that the concentrations used in Australia for fluoridation. Well, again, in that one sentence, they demonstrated that they didn't know anything about toxicology because they're confusing, once again, concentration and dose. And it's possible for somebody, even at 1 part per million, to get a higher dose than some people at 4 parts per million if they drink a lot of water. But be that as it may, it also shows that these people, these wonderful people selected by the Australian government and paid a lot of money, taxpayers' money, to do this report, which gave a rubber stamp to fluoridation safety and effectiveness, as they're about to do again in the next year or 2. Watch out, they're all set to do it again. It shows they didn't read, because if they'd read this report and got to chapter 2, there is a graph there. And in this graph, that red line across is the safe reference dose, the EPA safe reference dose of 0.06 milligrams per kilogram per day. And that dot at the top, which is way above that line, is the dose experienced by a bottle-fed infant, a non-nursing infant. So if they'd actually read the report, it said, a bottle-fed infant in a fluoridated community of 1 part per million is exceeding the EPA safe reference dose. How can you then turn around and say this is not relevant for Australia? I wish that these people's names could go in a book somewhere, because they're not serving their country, they're not serving science, and they're not serving our health. So basically, the third thing we know, and I've alluded to this already, American children are being hugely overexposed to fluoride from several sources, and we know that because of this prevalence of dental fluorosis. There was a report from the CDC in 2010, 41% of all American children aged 12 to 15 had dental fluorosis. And there is another report now, a later report, which says it's over 50%. And this dental fluorosis is not all in the very mild condition, which is what they were expecting for 10% of the kids when they started. But 8.6% is in the mild, and 3.6% is moderate and severe. This is all our kids now, all our kids. Go the kids in fluoridated and non-fluoridated. We're getting fluoride from everywhere. But the point is this. And they should have known this in 1945. They should have known that it would be reckless to assume that when fluoride is damaging the baby's growing tooth cells, that it is not damaging other developing tissues, like the bone, the brain, and the endocrine system. They can't deny the dental fluorosis because we can see it. But what about the things that we can't see? Those are the things that you have to study. You have to get government money, and you have to get scientists to actually study these things. And that is what they haven't done. They haven't done it in Australia and New Zealand. There have not been very many studies looking at the health of communities, looking to see if there's any other harm being done to children. Again, a disgrace. So, to the crux. The weight of evidence from many animal and human studies is that fluoride is a potent neurotoxicant. It can interfere with brain chemistry. Now, if you want to find out, I'm going to go through this now, I'm going to give you a lot of examples of the literature. But if you want to do it yourself, this is the way you do it. You go to our website, fluoridealert.org, /issues, /health, /brain. And you get all the studies on the brain, they're all listed, all the references are listed, all the IQ studies are all explained, every single study is explained what they did. Now, the reason I've spent some time on that, you can substitute 20 other words. You can put it there, bone, you can put teeth, you can put kidney, you can put liver, you can put any human tissue there, and you're probably going to get the same treatment in our database that you get for the brain. But for the moment, I would say if we can win the argument on neurotoxicity, it's over. It's over. So start with that. I just want you to know that everything you're about to hear is documented. So let's start with the animal studies. What have the animal studies found? Over 100 animal studies have found that fluoride damages the brain. It damages the hippocampus. It causes neurodegeneration. It inhibits cholinesterase, which is an enzyme involved in neurotransmission. It damages the nicotinic receptors, also involved in neurotransmission. It decreases brain glucose utilization. And it increases oxidative stress. It's just 100 animal studies. Now, I mentioned that I got involved in 1996. In 1995 was when Phyllis Mullinex published the first animal study, or at least in the West, the first animal study in the West on animals and the brain and fluoride. She looked at behavior. She had a model system using computers which can tell the difference of whether the animal was hyperactive or hypoactive, overactive or underactive. I won't go into the details. And she found that rats, if they were exposed to fluoride in the womb, before they were born, they were hyperactive. And rats who were exposed after they'd been born were hypoactive, underactive. When her study was published, she was told that her work was no longer relevant for dentistry, and she was fired. She was fired from the Forsyth Dental Center. So that was happening 1 year before I got involved. And then in the year I got involved, 1996, she was asked, Phyllis was asked in a press conference in Washington, D.C., were they interested in her study? She was asked in Washington, D.C., she said, do you realize what you're saying? You're saying that fluoride may be lowering the IQ of children. And she said, yes, I suppose I am. And the next year in China, the first 2 studies on IQ appeared, and sure enough, fluoride lowered the IQ of children. So that's an interesting bit of history. Not least is the politics, that she got fired for doing this. Now, I like these studies, I think, are particularly interesting. The good thing about animal studies is you can control everything. You can control all the variables except, in this case, giving fluoride to one set of animals and not to the other set of animals. You can't do that with human beings. You can't control everything. It's very difficult. But in this case, there have been 30 animal studies when they put these animals in different mazes, and the animal is taught that there's like cheese at the end of one of these tunnels, and so they could find out how well they learn. And it turns out that the animals that get exposed to fluoride learn less easily and memorize less easily than those that are not exposed to fluoride. So 30 out of 32 studies have found that in different types of tests, the Y maze, the T maze, the Morris water maze, novel object recognition test, open field test, and using low doses. The doses in these, this particular experiment by Lou is 5 parts per million. That may not seem low to you, but it takes at least 5 times as much fluoride to get the same serum level, plasma level of fluoride as a human being. And that's what's going to hurt you, is what level you accumulate in the blood, which circulates and delivers the fluoride to each tissue. So this will be equivalent to 1 part per million in human beings. Some more tests. Let's look at some more. They've got some interesting tests for young children. This is not IQ tests. These are very interesting. I never heard of this. My son was the one that taught me this. This one test, the Ray-Ostrich complex figure test. So, okay, so you've got this figure with all these elements in it. They present it to a young child. These are young children, and they say, copy this. So with this in front of them, the kid tries to make a copy. That's the first step. The second step is to remove the picture and say, draw it from memory. And it turns out that the children, or I better go to the official thing, the children who were given fluoride this is a high-quality study. They controlled for key variables like education, nutritional status, exposure to other contaminants, etc. They measured fluoride on an individual basis, the urine fluoride levels. And then the findings, approximately 9 out of 10 children were unable to copy this picture as expected for their age range. 9 out of 10 did not do as well as they would expect from their age. And then for immediate recall, almost 6 out of 10 were unable to draw the figure as expected for their age. Are you counting here? So we've got like biochemical tests, which shows that fluoride enters the brain, interferes with this, that, and the other. We've got animal tests for behavioral tests. We have here these learning and memory tests in animals. Now we've got this learning and memory tests in humans. And so now we're ready for the IQ studies. The current tally. Right now, there have been 50 IQ studies. 43 of them have found an association between fairly modest exposure to fluoride and lowered IQ. And these studies have been done largely in China, some in India, some in Mexico, and some in Iran. And here's a shocker. 19 of these studies, the fluoride water concentration was less than 4 parts per million, which is the EPA's safe drinking water standard. So when they say that these Chinese studies are not relevant to fluoridated communities, look at that. It's less than our so-called safety standard. I'm going to come back to that safety standard because the NRC recommended that that should be lowered in 2006. And here we are in 2015, and they still haven't lowered that so-called safe standard. But more about that in a moment, because that's another place where politics collides with science. Now, the public first became aware of these IQ studies in a big way when this review, a meta-analysis, sorry about that, there should be MET, meta-analysis of IQ studies. They took 27 of these studies, I think 25 of them were from China, 2 were from Iran, and they put them into one box, because they all had the same methodology. They compared the IQs of 2 communities, one with high fluoride, one with low fluoride. And what did they find? Of the 27 studies, this is in 2012, and the team included Philippe Brangin, who's one of the world's experts on mercury's neurotoxicity. He's just written a book called Only One Chance. He published a meta-analysis of 27 studies, where, because it was the same methodology, they added up all the data into one giant conclusion. And the study was published in one of the leading environmental health journals in the world, Environmental Health Perspectives. And they acknowledged that there were weaknesses in many of the studies. However, they also stated that the results were remarkably consistent. That even though they were carried out by different research teams in different parts of China and in different countries, the results were remarkably consistent. 26 out of the 27 studies, the IQ, average line IQ, was lower in the high fluoride village than in the low fluoride village. As I said, the world has heard about this. And the average lowering of IQ was 7 IQ points. Now, we get to a very interesting little quirk. In the press release that Harvard University put out, they made a mistake. They said the lowering of IQ was half an IQ point. And the proponents jumped on that and broadcast everywhere. It's only half an IQ point. This is nothing. This is nothing. This is within the realm of experimental error. But Harvard press people have made a mistake. It wasn't a half an IQ point. It was half a standard deviation. And half a standard deviation is about 15, 14 or 15. Half of that is 7 IQ points. That's the reality. Now, just to show you how propaganda has taken over our health agencies. A few months ago in New Zealand, there was this prestigious blue ribbon reports from Professor, what's his face, who was the chief scientific advisor, Sir Peter somebody, not you, Sir Peter somebody, Glickman, chief scientific advisor of the Prime Minister of New Zealand, and Sir Richard Skegg, the president of the Royal Society of Science, or whatever, in New Zealand. Very prestigious people. And when you get to the summary of the neurotoxicity data, now this is 2 years, 2014, and this study was done in 2012, what did they say? The lowering of IQ was less than 1 IQ point. In other words, they repeated this mistake from 2 years ago, which had already been cleared up in the media. And what does this tell you? These prestigious people didn't read the studies that they were concluding and telling the New Zealand population. That is disgusting. That tells you in New Zealand you can't trust these big names. When these big names come out and tell you something, you shouldn't trust them, because they've probably been told to do that if they want to get their whatever they get. Fascinating. Now, the other thing that the promoters said, they said, oh, there were methodological weaknesses. I agreed, but some of the studies were pretty strong. They weren't all weak. But the other thing they said is the concentrations in the high fluoride villages were very high and irrelevant to water fluoridation. So why are we going on about this? Okay, let's have a look at the 27 studies. Actually 20 of them, because about 6 or 7 of them were either done with coal or they didn't report the data. But there are the concentrations in the high fluoride villages. Look at them. 4.55, 0.88, 2.17, 1.8, 2.97, 2.11. Now what did the promoters do? They said, oh, these concentrations went up to 11 parts per million. They picked out two studies in which the range, in this one, 3.8 to 11.5, that one 2 to 11, and they took these out as if this was typical of those studies. Well, how typical are those numbers in this study? And if you've got a range, what you should be looking at is the mean. The mean of that is 7.65, the mean of that one is 6.5. But there's another thing here. If you're a toxicologist and you're presented with a lot of studies, and you're trying to determine whether they're relevant to the human population or not, or your population, what do you look for? Do you pull out the highest numbers, the highest doses, and say, you don't have to worry about this because it went up to this high dose? Or do you look for the lowest doses that cause harm? If your job is to protect the whole population, you have to go for the lowest dose. And that's standard. The lowest observable adverse effect level. It's a basic thing that you look for. So here is more evidence that the people that are making the rules on this toxic substance have no idea of toxicology. They don't know the difference between concentration and dose, and now it's clear they have no idea of how you go about risk assessment. They don't acknowledge that 8 studies were less than 3 parts per million. If you include those reporting a range of values, then 11 studies were less than 3 parts per million for some children. And if you take the low end of the ranges, then 16 had some children who were exposed to had lowered IQ, exposed to less than 3 parts per million. These are the 5. We have attempted to do a risk assessment, a health risk assessment. More about that in a moment. But we just picked out these 5 studies because these are all less than 3 parts per million. And so we have used this to say, well, based upon these results, what will be the safe dose? Because we're going to get to dose in a moment. What is the safe dose to protect all the children in Australia or New Zealand or America from fluoride's neurotoxicity? Based upon these findings. Okay. I want to give you more details on one study, IQ study, and that's the study done by Zhang in 2003. It's a particularly strong study. He controlled for arsenic, he controlled